Okay, good morning, good noon, good afternoon, and Assalamu alaikum and welcome to everybody. So I welcome on behalf of APPNA Merit Committee to all of you, and I'm going to introduce you to the speaker for today. And we are very lucky that we have Dr. Masood Bhatti. So I was so eager to listen to him for a very long time about his views. And then we have the wonderful panelist, Dr. Ubaidullah from the Drug Regulatory Authority of Pakistan, Dr. Abdul Rashid from the Drug Regulatory Authority of Pakistan, Professor Dr. Raza, who is in charge and, among the pioneer of having the bioanalytical lab in Pakistan. So one by one, I'm going to introduce all of the panelist and speaker to you. And please feel free to ask or write any question in the question answer session. I am Dr. Uzair, and most of you are familiar with me because we are running this series of webinar for a very long time. But for anyone who is not introduced to me, by qualification, I'm a medical doctor, but then I'm having a strong interest in pharmaceutical, vaccine, and biological. And I'm really lucky. I had an opportunity to see the working of Drug Regulatory Authority of Pakistan very closely as a member, expert member on biological and vaccine. So why I was so much intrigued about this topic, the reason being when I was medical, fresh medical graduate, and we received the patient, and some of the patient, they have some medicine which they have brought from US, UK, and they share their experience, very innocent people, that you see when we take this medicine, we feel better effect. Of course, that was the qualitative statement. And when we take the medicine from the local, it's not having any comparable effect. So I used to think that maybe it's a placebo, it's a psychological, but that was question always coming to my mind. So our medical colleague from US or North America, they cannot understand these issues. The reason being because here the Health Canada and the FDA and MHRA are a very strong bodies, but we have the evolving system. We are a developing country, so all the developing countries, they have their own share of all the issues, whether it's regulation, the quality, and the commitment from the industry. So I hope I'm going to find the answer to my question that about the generic medicine. So let me introduce Dr. Masood Bhatti, having a very, very strong footprint in the drug development ecosystem. So Dr. Bhatti has extensive experience in the areas of bioequivalence, clinical trials, pharmacokinetics, bioanalytical, pharmacology, toxicology, regulatory affairs, and project management. So he's a senior pharmaceutical research and development scientist with over 23 years management experience in planning, organizing, and execution of all phases of pharmaceutical development with leading multinational pharmaceutical organization and CROs in US and Canada. So after completing his B Pharm from Pakistan, he completed his Ph.D. in pharmacokinetics from the University of Alberta, Canada. He is currently working as a chief laboratory operation at Alpha Healthcare, Toronto, Canada. And before that, he was working as a chief operating officer at Biopharma Services Canada, director clinical operation at BioWell Canada, group leader at Purdue Pharma, New York, US, and study director at Covance Laboratories, Madison, Wisconsin, USA. He has conducted more than 700 clinical trials and coached team of clinical scientists to conduct and monitor high quality bioequivalence phase 1, 2, 3, and 4 clinical studies. So he's a pioneer scientist who transferred clinical trial knowledge from North America to Pakistan among the pioneers and helped establishing one of the first bioequivalence clinic in Pakistan. He has numerous peer-reviewed publication and international presentation to his credit and has successfully hosted multiple regulatory audit across the world in his eminent career. So that was a very, very brief. He has much more to his credit. Let me introduce the panelists. So first of all, we have Professor Dr. Muhammad Raza Shah from ICCBS University of Karachi. So he's a professor over there at HEJ Research Institute of Chemistry, University of Karachi. And he's also heading the Center for Bioequivalence Studies and Clinical Research, head of Nanotechnology Center at ICCBS University of Karachi. He holds his PhD from Max Planck Research Institute, Germany, and postdoc from Geneva University, Switzerland. He has published more than 400 research articles in international journal with cumulative impact factor of more than 1300 and published 5 books and 12 chapters by elsewhere and 2 US patents. So he also received his Tamgha Imtiaz from President Islamic Republic of Pakistan, received Top Scientist Award from KPK government, and many more, and selected as the Third World Academy of Science Young Affiliate and Fellow in International Union of Pure and Applied Chemistry, Chemical Society of Pakistan, Academy of Sciences, and Editor General of the Chemical Society of Pakistan. And our dear friend from the Drug Regulatory Authority of Pakistan, Dr. Abdul Rashid, he is looking after the pharmacy services and where we have these bioequivalence studies and all these issues. And our old and dear friend, Dr. Ubaidullah, he's a secretary registration board and who is deeply involved with the registration of generic medicine. So these are the expert and the champions of their field. So I think I'm so excited with this webinar where we have the best people from academia, from the technical people, from the regulator, and we are trying to find the answer to the question that if we prescribe the generic medicine, are they the same like the innovative product? So that is the key question. Being the prescriber and the user, I mean, the medical community, they just follow the guideline from the regulator. But, it's a consistency in the North America and other stringent regulatory authority markets. But for Pakistan and other developing countries, we always face this challenge. And it's a deep-rooted issue, and there is no ideal solution. But we are struggling to move in that direction. And we have the representative from Drug Regulatory Authority of Pakistan, who after the discussion and the talk, share their experience and their commitment and vision in that direction. So with this, I really request Dr. Masood Bhatti to start his presentation.
Thank you, Dr. Syed Uzairul Ghani. I'm very humble with your detailed introduction. Thank you so much. And thanks to the panelists, Dr. Abdul Rashid, Dr. Abadullah, and Professor Dr. Raza Shah. And I would like to thank everybody who has tuned on this Saturday morning in North America and in Pakistan at 9 o'clock. Thank you so much. So, as Dr. Uzair mentioned, the topic is generic drugs. And there's a lot of myths, mysteries, and we'll try to discover or try to find what are the actual facts. I just wanted to put this disclaimer that during this presentation, different aspects of generic drugs will be discussed. Both legal and medical aspects of generic drugs will be presented with the purpose of education only. Nothing stated during this presentation should be considered as medical or legal advice. If you have medical or legal concerns, please consult a doctor or a lawyer. Although my research has been carried out to the best of my knowledge and ability, this presentation is issued without any legal liability on my part. I have no conflict of interest regarding this presentation. So what are the common questions? The common question, what is a generic drug? Are generic drug the same as the name brand? So we will talk about, we'll touch base a little bit about name brand drugs, then we will focus back to the generic drugs. But because we're comparing generic drugs with the name brand, which are the innovators, or sometimes they are called also NCE. You will hear this word throughout my presentation, new chemical entity. Are generic drugs safe? Are generic drugs less effective? So this is, I've been asked, like as a pharmacist also when I was practicing pharmacy, that are the generic drugs less effective than the name brand? So why are generic drugs cheaper? So if they are the same, if let's say if they're the same effective, then why they are cheaper? And are generic drugs work the same way? So this is a little bit more pharmacological question, that as their mode of action or mechanism of action is the same as the brand name. So during this presentation, I will try to answer some of these questions. So some background information regarding the brand name drugs, or the new chemical entities, or NCE. So to understand how the brand name drug comes to the market, I'm going to briefly touch base the drug development process for new chemical entity. So the research, the great minds, professors in the universities are in the pharmaceutical research centers and R&D, they are continuously trying to discover new molecules. And as you understand that based on the pharmacological concept of receptor theories, they are trying to understand how to best get a key, lock and key mechanism which can fit on that particular receptor, which will trigger the pharmacological action. And these are all in vitro studies, and it has been continuously taking place in the leading pharmaceutical research centers, as well as in the universities, in the pharmaceutical department, pharmacy departments, and other research centers. And after 3 to 5 years, thousands of molecules are screened, and then few of them are nominated for the preclinical research. So because this is a new chemical entity, it has never been introduced to human being. So we start with animal studies first. So these are basically the safety studies to understand the pharmacokinetic and toxicology of the drug before we enter into human. Preclinical studies, also called IND submission studies, or investigational new drug studies, take about 1 to 2 years to do that. And once the animal safety is established and the in vivo theoretical proof of concept is somewhat answered in the mammals. Like you start with the rodent and then we go to mammals in animal study, preclinical studies. Then we submit IND to regulatory authorities. And after ethics review board approval, the molecule is nominated for phase 1 studies. So phase 1 studies are mostly, as you all know, they are safety studies. We are not looking at the efficacy or pharmacological action. of the drug at this stage. We just want to make sure this is a new chemical entity, has never been introduced to human, and we are very, very concerned. So these studies are done under hospital, just like ER, emergency room type of setup, where you have paramedics on hand, and if there is allergic reactions to handle those side effects, we have a team of nurses and technicians on hand. And due to based on the pharmacokinetic knowledge which we learned during the preclinical research, we do what we call it dose scaling, based on the mathematical models. And whatever the dose comes in, we go with the hundreds of times lower than that dose, and we start with the cohorts. So this is called phase 1 studies. So the safety is the main concern here. So when the safety is established, we gradually increase dose for the second cohort, third cohort, fourth cohort. This is all within the phase 1 studies. Once the safety of the drug is established, then we go to a very small number of subjects. So these all studies so far, phase 1 and phase 2, are done into normal healthy subjects. We are not talking about patients. We didn't talk about pharmacology or efficacy at all. So we are concerned about the safety because, as I mentioned, this is a new molecule, and we look at its pharmacokinetic profile. That the whole idea is the drug goes into the body, and then it should be excreted. So we do mass balance studies. We try to understand what is its mechanism of excretion. Does it excrete it through kidney or liver, or what are the metabolites, and are the metabolites are safe or toxic? And so all these things are done between phase 1 and phase 2 clinical trials. Then we go to phase 3 clinical trials. Here we start with efficacy studies. So now the safety is going on, but phase 3 studies are done in patient. So now we have a disease model. We understand what is the endpoints are. So the protocols are a little bit more complicated, more bigger, and the number of subjects, number of patients in this case are also larger. There's a multi-center clinical trials are done. So this whole process in clinical research, starting from phase 1, 2, 3, it takes up to 6 years. And then the data collected from all these clinical research is submitted to the regulatory agencies. And because it takes a long time to do these studies, so we have a large number of patients' data available. So it takes about 1 to 2 years to review. And once the review is done, then the drug is approved. Drug is marketed with caution that there will be phase 4 clinical trials, which is called post-market drug safety monitoring. So even though now the drug is legally available in the market, we can sell it, physicians can prescribe it. However, there is post-marketing safety monitoring. There is a 1-800 number. So physicians and pharmacists and even patients, they're all encouraged and required that if they see any type of side effect after the drug launch, they call the drug company, and the drug company is required by law to inform this to the regulatory agencies. So from the lab bench side to the bedside, it takes almost 10 years and close to $1 billion to bring one new chemical entities in the market. So that's why you won't see there's a lot of drugs coming in, an average less than 20 and sometimes 15 new chemical entities are approved in North America. There are other drugs which are approved, which are called 505(b)(2), but those are not new chemical entities. There's a lot of other reformulations, control release, sterile release, oral disintegrated tablets. So those are not new chemical entities. All those drugs are reformulating of the existing molecules. So once this drug is now in the market, before I go to that, I'm just going to summarize this, whatever I have said this now. This is, I got this from the FDA Center for Research. There are 10,000 compounds it starts, which are synthesized and purified in the beginning. And out of them, 250 compounds are nominated for animal testing. Out of 250 compounds, only 5 compounds reaches to the clinical studies, which is phase 1. So there is a phase 1, phase 2, and phase 3 studies. So the compounds get killed either in the phase 1, phase 2. In the end, only there is one compound which is approved and it hits the market. So you can appreciate and understand that how difficult it is and how lengthy, how costly it is to bring a new product in the market, the brand name drugs. So that's why these brand name drugs got exclusivity. So they got patent. So once this drug is in the market for the number of years, nobody else can copy this drug. So that's why you cannot see the generic drug right away. So generic drugs are not new chemical entities. We have to understand that. The generic drugs are the drugs which have been in the market for a long time. So as you can see with the drug development process, it takes about approximately an average about 6 to 10 years of aggressive and detailed testing of one new chemical entities to hit to the market. And during the drug development process, all the research is done in a controlled fashion, and then the drug goes into the market, and now it is prescribed to millions of patients for several years, several years, depending on the patent period. Brand name drugs have several years of exclusivity. Generic appears in the market after the patent is expired. So generic is done through ANDA, whereas the new chemical entities or brand name drug is done through new drug application. With the new drug application, I have explained in my previous slides that we start with in vitro, animal, phase 1, phase 2, phase 3, then post-marketing, and then submission and approval. Whereas abbreviated new drug application, which is ANDA, this does not require any toxicology studies. It does not require any animal studies, because as you can see that the drug molecule is already in the market. It has gone through phase 1, phase 2, phase 3, controlled clinical trials, and then it had gone through the public use for several years. So there is no need to do any further toxicology for that particular molecule. The efficacy studies is also not required, because this drug has been studies exclusively for that particular indication, for that disease, and it has been prescribed, and we know what can be the expected adverse events, side effects. So we learn all this through our drug development process, as well as the several years, which is sometimes more than 10, 15 years of the patent period for the brand name drug. So abbreviated new drug application requires, I'm going to touch that when we will discuss about bioequivalence and the generics. So it's important to know that the generic drugs does not require new drug applications. It required only abbreviated new drug application. And also there's no IND required for generic drugs. IND meaning is all the animal studies. So now we go to some of the terminology, touch base about what are the pharmaceutical equivalents. What are the pharmaceutical alternatives? Therapeutic equivalents. And what is the bioavailability? To understand bioequivalence, we need to understand what the bioavailability is. So pharmaceutical equivalents are the drugs, products are considered pharmaceutical equivalents if they contain same active ingredient. So this is very important. So the active ingredient has to be the same. same limit of impurities. So this topic, this thing usually is forgotten. You know, I don't want to say it's usually forgotten in our countries because we focus mostly on the active ingredient, but there are some impurities. Those impurities limit is defined in the compendial books, in the United States Pharmacopoeia, British Pharmacopoeia, European Union Pharmacopoeia, so name it. So the impurities has to be the same in the generic, in the pharmaceutical equivalents. I didn't even arrive to the generic yet. So let's talk about pharmaceutical equivalent first. Same dosage form, same route of administration, identical in strength. So equivalent products contain the same amount of ingredient if the same dosage form, but may differ. So now pharmaceutical equivalents may differ in characteristics just as shape, release mechanism, and packaging. Now we have two products here. One is a test product and other is a reference product. So let's say reference product is your brand name drug and test product is your generic drug. So we said, okay, they are both pharmaceutical equivalent. I think this is, I wanted to focus on this for a little bit, and I will request everybody's attention here, because here where we because I work in the health system in Pakistan, have been working in North America for last so many years. We consider pharmaceutical equivalents if, as long as the reference and the tests are pharmaceutical equivalents, we think that, you know, they are good to go. However, there may be some possible differences. So drug particle size. So particle size is important because when we go to the classification of biopharmaceutical drug classification, you will see how important it is. Excipients, so can be different because in the pharmaceutical equivalent, they can use different excipient, and excipient is very important for disintegration and dissolution of the drug tablet into any solution, and it dictates the release mechanism, and which ultimately dictates your Tmax and Cmax and eventually the therapeutic effect. Manufacturing equipment or processes can be different, site of manufacture can be different. So all these differences could lead to differences in product performance in vitro. So even though these are pharmaceutical equivalent, and pharmaceutical equivalent is a big deal, like we went through, like they have to be the same strength, shape, the same. But however, even though the drugs, if they are pharmaceutical equivalent, they may differ. This is just, I just added here that there can be pharmaceutical alternatives, but this has nothing to do with the generics, but I thought it's interesting to show what a pharmaceutical alternatives are. What are the therapeutic equivalents? The drug products are considered therapeutic equivalents if they are, number one, they have to be pharmaceutical equivalents. Number two, they have to be bioequivalents. And they are approved as safe and effective, and they are adequately labeled, manufactured in compliance with manufacturing practices guideline. Now, therapeutic equivalents, which are now, I will say these are the generic drugs, they are expected to have the same clinical effect and safety profile. Now, I'm going to highlight that they are expected to have the same clinical effect and safety profile because they have the same ingredient, and for the safety profile, they have the same limit of impurities, but they are expected to have. So that's why I think just consider this as I further go into my presentation, then you will appreciate that why I highlighted these. So the bioequivalence drug product are considered bioequivalents if they are pharmaceutical equivalents. The rate and extent of absorptions are not statistically different when administered to patient or subjects at the same dose under similar experimental conditions. And these experiments are called bioequivalence clinical trials or BE clinical trials. So when we talk about BE clinic, those bioequivalence clinics, they perform bioequivalence studies to see that if the test is statistically not different from the brand name. Bioequivalence may be demonstrated through in vivo as through clinical trials or pharmacodynamic studies. So in rare cases, there is a pharmacodynamic studies are also done to show the bioequivalence. So there is a strong relationship between single dose pharmacokinetic and therapeutic effect, but however, there are some assumptions. So we do all these clinical studies, bioequivalence studies, but they are all based on some assumptions that the single dose pharmacokinetics are similar to steady state pharmacokinetics, because in patients, we don't give a single dose. They take like dose for several days, several times a day. Patients have a pharmacokinetic profile compared to that of healthy volunteers. The bioequivalence studies are done in healthy volunteers. They are not done in patients. And we are assuming the pharmacokinetic and bioequivalence studies are all PK. They're all pharmacokinetic studies. And I'm going to touch base some pharmacokinetic parameters which we compare. What we are saying is that the patients have the same pharmacokinetic profile, which is compared to healthy volunteers. So these are some of the assumptions. And then minus 20% to plus 25% difference in the absorption, which is Cmax, and area under the curve has no effect on therapeutic outcomes. So this is really interesting. And I thought I should bring it in, even though, this is a regulation states that if the brand is minus 20% to 100 and plus 125%, they are within that range. They are equal. However, I wanted to draw attention that there is a big range, and this is important for highly variable drugs. So we talk about oral dosage form, that drug has to be released first into the solution, into the stomach. So that's why excipients are very important. So dosage form has drug go into the solution, then it has to cross the drug wall, then it goes to the blood, and there we measure the pharmacokinetics. So all the bioequivalence studies are basically comparing the relative bioavailability of two products. And so that is the pharmacokinetic, whereas the after drug goes into the site of action, and then it produces the pharmacological effect, which are now clinical or PD measurement. So this is the biopharmaceutical classification system for the drugs. Here you can see there are 4 classes of drugs. 1, 2, 3, and 4, simple. And they all depend upon the solubility and the permeability. So if you go to the previous slide, you can see the drug has to go into solution first. So what that means, the drug has to be highly, highly soluble in water. But then what? Then it has to pass the gut. It has to pass the gut wall. And gut wall, you need to have some kind of lipid penetration, because gut wall has some fat in it. So you can appreciate that how difficult it is for the synthetic chemists, that first of all, they have to come up with the compound which is matches with the receptor as like a key and lock type of theory. But then they have to make sure that this drug is highly soluble. But at the same time, it's a highly water soluble, but at the same time, it has lipid permeability. So that's why class 1 drugs, which are highly soluble and highly permeable drugs, they are a small number of drugs. And for these type of drugs, the class 1, you don't need to do any bioequivalence study. You don't need to do any BE study. However, you have to file a bio waiver to establish through different literature documents and your own studies that your drug is highly soluble and highly permeable. The second, third, and fourth classes of drugs. Second has a low solubility, but it has a high permeability. Third one has a high solubility, but it's a low permeability. And then class 4 is actually really poor. It has a low solubility and low permeability. So all these classes of drugs require bioequivalence studies. So as I mentioned, we are talking about oral solid dosage. There is no bioequivalence studies required for injectable, IV and solutions. And the solid dosage form, which are class 1. However, you have to still file a bio waiver. So what are the comparative pharmacogenetic studies? So it is in vivo measurement of active ingredient. So some relationship between concentration and safety and efficacy. Product performance is the key. So the comparative bioavailabilities are done through bioequivalence studies. So what is bioavailability? The rate and extent to which a substance or its active moiety is derived from the pharmaceutical form, like tablet, and becomes available in the general circulation. So as I mentioned, intravenous administration has 100% bioavailability. So there are drugs in the market which has as low as like 20% bioavailability. Bioavailability actually is, if I go a little bit more, that not only that it is limited or governed by the dissolution in the solution, but also the penetration of the gut, and after that the liver, because that's the first pass metabolism, because everything which goes through the portal vein into the liver, and then if it's a highly metabolite drug, even though it is highly soluble, highly permeable, still it's what it's called first pass metabolism. It can decrease its bioavailability up to 80%. The example is verapamil. Now, bioequivalence studies. It is in vivo measurement of active moiety or moieties, drug and active metabolized in biological fluid after single dose of test and reference product. And what are the important pharmacogenetic parameters we're going to compare? Area under the curve, AUC. Usually it is AUC 0 to tau. Measure of the extent of bioavailability. So AUC will tell you that bigger the area under the curve, higher is the bioavailability. Cmax, it will tell you the onset of action, like how soon or what is the observed maximum concentration of drug. A measure of both the rate of absorption and the extent of bioavailability. The Tmax is the time after administration of drug at which Cmax is observed. So it's the onset of action, because what happens is that after oral dose administration, drug goes into the solution and then gradually it passes through the gut wall and goes through if it survives liver, and then it's gradually start getting available at the central circulation system, systemic circulation system. And the sooner it reaches the site of action, the sooner it will be the onset of action. So that's why, the onset of action really determines how fast the drug absorbs. So that's the Tmax. So definition of bioequivalence. Now we talk about bioavailability. Now what is bioequivalence? Pharmaceutical equivalents, whose rate and extent of absorption. We talked about rate and extent. Rate is like Tmax, extent is the Cmax, are not statistically different when administered to patients or subjects at the same molar dose under similar experimental conditions, which are bioequivalence studies. So what is the purpose of bioequivalence study? So therapeutic equivalence. So bioequivalent products can be substituted for each other without any adjustment in dose or other additional therapeutic monitoring. So once we establish that the product is bioequivalence. There is no need to establish any therapeutic equivalence. There's no need to do any clinical trials for efficacy trials. So this is a typical plasma concentration time profile, which tells you, which shows like Cmax, Tmax, and AUC. I'm pretty sure you have seen it so many times, hundreds of times. So AUC dose relationship, linear pharmacokinetic. I talked about linear pharmacokinetic, and in the study design, you will see why it is very important, a linear pharmacokinetic, and if it's a saturation, and if it's non-linear pharmacokinetic, then the study design may need to be revised. We need a different study design. And here you can see as the dose is increasing, so dose versus plasma concentration, which is basically in this case is the AUC. So as the dose is increasing, the proportionally AUC will increase. So that's called linear pharmacokinetic. And of course, it's up to certain limit, and after that, it will go to saturation. So for a bioequivalence studies, let's talk about study design. So single dose, two-way crossover, fasted. This is the most desirable study design for a typical bioequivalence studies. So it's a two-way crossover and fasted. And fasted because the drugs may exhibit a food effect, like there may be interface in the presence of food. However, there are studies which are done fed. It's a single dose, two-way crossover. But these studies are done for those products where the indication for the brand name drug have a label instructions that it should not be taken empty stomach, like for example, NSAIDs. So other study designs are single dose, parallel design, fasted, and this is for the long half life studies where the washout is a problem. It's a very, very long half-life drugs. Multiple dose, two-way crossover. Multiple dose are done when the HPLC assay or LC mass spec assay, the bioanalytical assay is not sensitive. Like there is, the technique requires that, you need a accumulation of the drug in the body before we start collecting it. So we do multiple dose. Clinical endpoint studies are done in patient. And these are done only for those products where the plasma levels are not adequately available, or the site of action is not in the systemic circulation. For example, nasal decongestant, where the site of action is in the nasal mucosa cavity, and then you just spray and the drug acts there, and very little is available in the systemic circulation. So you cannot, collect enough, you cannot detect enough plasma levels of the drug in the systemic circulation. So in that case, it's required to be clinical endpoint studies need to be done, which are very expensive, very long. And for the purpose of this presentation, I'm going to focus only for the oral dosage forms. However, I thought I should bring this into this equation also, that the bioequivalency also be established through clinical endpoint studies where plasma levels of the drugs are not easily detectable. I talked about the sample size. So the variability of the drug is very indicative of how many subjects you will need for a bioequivalence study. So if the variability, inter-subject variability is low, it's a normal variability. For example, I gave you an example that from 80% to 125%, a low variability, a normal variability for the certain number of subjects, the study will pass. But if it's a highly variable drug, you may need to increase the size, the sample size. And statisticians in the audience and the scientists, they will know this more. that, you know, how it is very important that the higher the variability, intra-subject variability, the larger number of sample size you need to show a statistical difference between two products. There's a multiple dose studies may be employed when there's a non-clinical pharmacogenetic saturated metabolisms. Drug measurements technique is not sufficiently sensitive for single dose, which is a high performance liquid chromatography assay or LC mass spec assays. Crossover parallels design is also preferred when there is an intra-subject comparison, low variability, generally fewer subjects is required. So the crossover is the preferred, as I mentioned earlier, design. It is preferred because the subjects are acting their own reference in this case, because you're not getting another set of subjects to do the comparison of the drug. So the subjects come in and we randomize them into two groups. One group gets the test, other group gets the reference drug, and then we collect their blood samples, and then they go home after 2 days, and they come back after their washout period, and then we do crossover. So it means that the subjects who received reference drug, they will receive now the test drug, and the subjects and the vice versa. So it means that the subjects itself, they are acting as a reference. So we are not introducing any other variability. So generally fewer subjects are required. However, in certain cases, crossover design is not usable. Maybe, as I mentioned, drug has a very long half-life. So then you have to have two groups of subjects starting parallel. So its crossover design is, in this case, is not usable. So it's very important to make sure that our statistician has done their subject size determination using their statistical method based on the intra-subject variability. And adequate samples are also collected. It's all depend upon you know, where your Cmax is. So we don't want to miss the Cmax, because if you miss the Cmax, you will miss a big chunk of area under the curve. So the study design is like you need a very strong pharmacokinetic to design this study, depending upon the pharmacokinetic profile of the innovative drug, and going through and collecting a lot of samples around the Cmax time point, and then making sure that we collect enough samples to capture the elimination phase, to make sure that we have the right number of samples to determine the half-life and the AUC. And completion of gastrointestinal transit and absorption process. These are all should be considered when to give them food and what type of food, when they can drink water. All these are controlled studies, so the subjects are given similar type of food, similar type of amount of water. If they are fasted, then they will not eat anything at a certain number of hours. This is obviously all we'll discuss with the ethics review board, and it will be spelled out in the clinical trial protocol, which will be submitted to the regulatory agencies as well as the ethics review board. Fed condition depend on local diet and custom. So let's say that we have to do a fat study. We cannot do fasted because it's gonna hurt the stomach. So it's very important that fat study, let's say if we're doing in Pakistan, we should do in the local diet. Like wherever we do this, we should not change suddenly, like because we're doing a fat study and FDA says you have to have this like this type of food. So we should consider as a pharmacokineticist that if we change suddenly the diet, the PK the drug PK will affect or may affect. So minimal meal to minimize impact required due to drug substance and drug form. Modified release products also required fat studies sometimes. Complicated pharmacokinetics, some drugs required fat studies. Known effect of food on drug substance. This is the big one. Like if we know that the drug is already in the market for several years and the label indicates that there is a food effect. So it should take with food or without food. So if there is something like that, it means that it's going to affect the pharmacokinetic of the drug. So in those cases, we cannot do a fasted study. So we have to do a fat study. And usually dosing is done in the morning. It is just logistically, it's more appropriate. So that's why in the fat studies, we give them high fat breakfast. It can be any type of breakfast. It has to be high caloric, warm breakfast for the fat studies. So in the statistical analysis, when we're comparing brand versus generic, we compare AUC and Cmax, and 90% confidence interval CI must fit between 80 to 125%. So the bioequivalence criteria is done through two one-sided test procedure. Test product is not significantly less than the reference product, and reference product is not significantly less than the test. So significant difference is 20%, alpha is set at 0.05 significance level. So it means T over R is 80 over 100, which is 80%, and R over T is 80. All data expressed as T over R. So this becomes 100 over 80, which is 125%. So the difference is between 180 to 125%. So what are the requirements for a generic drugs? So beside the bioequivalence, I touched how to do the study, how to do the study design. Very important thing is legal. We cannot bring a generic drug in the market until the patent is expired. So it has legal consequences. Labeling is very important. Labeling has to be the same as brand. Chemistry and microbiology is very important. So chemistry, meaning that all the chemistry, control, manufacturing, microbiology is the USP chapter 61, 62. It has to be free from all the yeast and mold and pathogens and total plate count and all these tests. It has to be meeting the requirement of a clean drug. So what are the requirements for a generic drug? So same active ingredient, same route of administration, same dosage form, same strength, same condition of use compared to reference listed drugs. So during the studies, we call it RLD. So RLD is listed in the Orange Book. So the US FDA has an Orange Book, which is the brand name product. They will say, like, let's say if a brand name product has several strengths, FDA will identify one strength, which will call the reference listed drug, RLD. And if we do an RLD to that strength with the generic drug, that will be considered, it is equivalent to all the other strengths, other label claims. So labeling is very important. Labeling has to be the same as brand name. We have to exclusively draw important attention to the adverse effects, expected adverse effects. So whatever is on the brand name, because we're not doing any toxicology studies, we have not doing any efficacy studies for the generic drugs. May delete portions of labeling protected by patent or exclusivity, may differ in excipients, PK data, and house supplies. Chemistry, we talk about it, that component and composition, manufacturing and control, batch formula and records, description of facilities, specs, and test packaging, and stability has to be done also to make sure the drug is stable, whatever the expiry date is listed on the drug. So we talked about the question in the beginning. So I'm winding up my presentation with trying to answer some questions. So what is a generic drug? So generic drugs are bioequivalents to the name drug. And we talked about how we reach to that bioequivalence. Are generic drugs the same as name brand? Yes. However, regulation allows minus 80 to 125% difference. Are generic drugs safe? Yes. Same active ingredients, same impurity levels. Impurities has to be the same. So, you know, these impurities are degradation products, related compounds when the synthetic chemists, you know, it's called API, active pharmaceutical ingredients. So when we do the API determination, we want to make sure the active ingredient as well as the related compound, the degradation products are the same, impurity level has the same. Because some impurities are very toxic. Are generic drugs less effective? No. However, I just put a question mark here that if drug is still approved, but it has only 80% AUC as compared to the brand name. We have not done the clinical efficacy study because it's not even required by law. So this is a question mark. So I'm going to leave this like this. However, the generic drugs are less effective? No, I would say. But if in practice, if somebody sees something is a little different, you know, it may be the drug has a little bit lower level, but I will not go further in comment because the drug is approved and it is allowed by the regulators that minus 80 to 125%. If they are different, they are still equivalent. Why generic drugs are cheaper? So they are cheaper because we don't have to file an NDA. It's not a new chemical entity. You don't have to go through the clinical trials. You don't have to do the toxicology studies. And you know, NDA takes about $1 billion to bring one new chemical entity into the market, and up to 10 years, 6 years to 10 years. Whereas every new drug application in North America can be done within less than $1 million and within less than 6 months. So that tells you that why generic drugs are so cheap. Are generic drugs work the same way as the name brand? Yes, the pharmacological action, the pharmacogenetic is the same because it has the same active ingredient. I want to thank you for everybody listening for so patiently. Thank you so much. And I will open the forum for questions.
Thank you very much, Dr. Masood Bhatti. Trust me, this is one of the best presentation I have ever attended. And being from the medical background, I think generally we know very little about the bioequivalence aspect, and we have very little technical know-how about this thing. And I think at the undergrad and the postgrad level, this should be part of the curriculum to the medical prescriber, particularly for the developing countries, that they should have a fair idea about the therapeutic equivalent, pharmaceutical equivalent, bioequivalent, and the role of impurities. Because trust me, my feeling is that from the safety perspective and the impurity, I think this thing needs to be considered quite seriously for developing countries. And but from the prescriber side, the medical physician, they have very little knowledge about many of these aspects. And we are lucky we have a lot of expert over here. So let's involve them. Dr. Raza, can you share your experience from your bioequivalence lab in Karachi University? What you have been doing, what your institute is doing, and what are the capacities in the country at this point in time?
Thank you very much, Dr. Ozair, and I would also like to thank Dr. Masood for such a wonderful presentation. So it was a student-like presentation. He made the most complicated topic so simple so that everyone can understand it very well. With regard to our center, so the Bioequivalence Center is an integral part of International Center for Chemical and Biological Sciences, ICCBS, which is also very well known by the name of HEJ. So it is a UNESCO Center of Excellence, and it is also WHO collaborating center. So it is considered to one of the premier research institute in the country with over 500 PhD students, 17 research building. So one of the research building is bioequivalence studies and clinical research. So we have two component in the clinical research. One component is just assigned for bioequivalence studies. So our center was among the first one which was approved by Drug Regulatory Authority of Pakistan for the clinical trial, and currently is the most active one. So far we have conducted a number of bioequivalence studies for multinational companies, which include Merck, for Pfizer, GSK, Abbott Laboratories, and also very good national pharmaceutical industries like Sami Pharmaceutical, PharmaView, and Hilton. So there is a lot of going on in the bioequivalence area in our center. Our center, like the bioequivalence facility, is one of the best in this part of the world. The covered area is more than 32,000 square yards, which is, and it is we can host at one time 110 healthy subjects. So this is quite rare to find in a bioequivalence centers anywhere in the world which is having the capacity to just accommodate 110 subjects at one time. We are the analytical facility of our center is as good as anywhere in the world. It is considered to be one of the best analytical facility in this part of the world. We having a number of HPLCs, LCM, SMS for pharmacokinetic studies. And in addition to that, our engineering system is extremely strong one. So all the analytical equipments which we are having, they are functional, and our engineering staffs are very well trained, and they are taking care of those equipments very well. Beside the bioequivalence studies, we are also actively engaged in clinical trials. We conducted the phase 1 clinical trial of Sinopharm vaccine, and then we also conducted a clinical trial on COVID-19 patient where we used the traditional Chinese medicines. And this was the medicines which were there in the national guidelines of China. And when we had a discussion with the ambassadors of China in Pakistan, so he and a team from the China recommended that we should conduct clinical trial on these traditional Chinese medicines and see its effectiveness on Pakistani population. So that's the trial which I led as a principal investigator from the platform of our clinical trial unit. Beside that, we also conducted clinical trials on bronchitis. We also conducted, and that is also like traditional Chinese medicines, which we compare with the Augmentin. These were double-blind randomized controlled trial. And then we also conducted clinical trials on gastritis and pelvic inflammatory disease, and a number of others are also pipeline. So the center is really well equipped. The staff which we are having in our facilities, they have also got training over the last 10 years. Our center became functional in the year 2010, and since 2010 till today, we are very actively working in the area of clinical trials and also the bioequivalence studies. So it is one of the premier research institution in bioequivalence studies and clinical trial. We are also conducting workshops and training workshops for the local pharmaceutical industries. Like I have conducted workshops on the bioequivalence studies in Lahore and in Islamabad and in Karachi, many workshops. We are also engaged with the Drug Regulatory Authority of Pakistan in various forums, like with the previous CEO, Dr. Aslam. I worked with him in this BioStudy Rule 2017, where we conducted meetings in our centers and he conducted meetings with the stakeholders in Karachi. So we and me and Dr. Abdul Rashid, we are also working closely in just arranging workshops in the clinical trials and training sessions. Like even alone in the last week and last 10 days, we conducted two. One was in the COMSTEC, this is for the OIC countries. And another one was also conducted in Lahore, in concerts universities, and where our doctors and clinicians and representative from the pharma industry and drug regulatory authority were there to attend these workshops. So we are working with Dr. Abdul Rashid. I'm closely working to just create awareness in this area. So that is all, and I'm
Great.
really thankful to everyone for this, giving me the
Great.
opportunity to talk here.
Excellent. So we have with us Dr. Ubaidullah. So Dr. Ubaidullah, can you tell us what, as a regulator, you have in the pipeline to assure the quality of generics available in Pakistan? And because as a prescriber, the doctor, they just prescribe, so they have no idea really whether the product is good or not, because everything, whatever is registered, they take it like that. So that is a very critical question, and that I'm also discussing with the policymaker, with the DRAP and the Ministry of Health, and all those levels. So I know that you are struggling very hard, and we have a lot of people from the industry in this webinar as well. I'm so happy and glad. Please share what you have in place really to move in that direction, please.
Thank you very much, Dr. Uzair, and I really appreciate Dr. Bhatti for very beautifully articulating the drug development process, both for new chemical entity, which we call innovator brand, and specifically generic drug product, which we call me-too drug as well. By and large, he has very clearly stated that the criteria for any approval by any regulatory authority for the innovator drug or new chemical entity is the safety, efficacy, and quality. And by and large, this safety parameters is mostly pertained to the new drug development and new chemical entry by the innovator drug. And the followed up companies who are manufacturing generic drug product, they are mostly concerned with the efficacy profile of the product, in part of efficacy profile, and then the quality parameters which they are supposed to follow. If we see that the efficacy, if we see that the clinical trial part of the efficacy is by and large, which is related to the generic drug product, that is the bioequivalence studies for new chemical entity and biosimilarity studies for the biological products. And at the same time, if we see that all the pharmaceutical or biological products, by and large, if we see the quality parameters, then the quality parameters for all these products have been clearly enlisted in the reference books, which we called pharmacopoeia. So if we follow that pharmacopoeial specification for the quality testing and also for the regulatory approval, then that by and large that covers the quality part of any regulatory process. Now come to the dynamics where the efficacy part and the bioequivalence studies are related for any regulatory approval. One thing I want to mention here that this bioequivalence study is by and large related to the OSDs, oral solid dosage forms, which are supposed to be taken by by the GI tract, and all the phenomena has been operated by the Dr. Bhatti. And in order to achieve all these things, after the establishment of GRAP in 2012, we follow most of the international guidelines for all the regulatory processes. And for this purpose, after hard work and struggle, you know well, we adopted a common technical document after so long effort by our regulatory authority, and we succeeded to implement common technical document as a registration application or marketing authorization application in Pakistan. And just to share with you that Pakistan is the first country of the region who has adopted common technical document as a registration application. And after Pakistan, other countries adopted that. And if we see this common technical document, so just to review that, it has 5 parts, module 1 to 5, and it covers all the parameters which the Dr. Bhatti has elaborated from starting from the CMC, chemistry, manufacturing, and control of the drug substance, up to the end part, that is the trial and putting the product up to the shelf life. But mostly in Pakistan, as most of our companies are generic companies, so they don't need to do the clinical studies, as already shared by Dr. Bhatti. So we are focusing mostly on the parameters of efficacy, which is related to the equivalence studies, and secondly, which is the quality parameters, which we are perfectly following after the adoption of this common technical document. As Dr. Bhatti has also shared that all these bioequivalence studies follow the BCS classification system, biopharmaceutical classification system, which have 4 classes depending upon the solubility and permeability of any drug substance and product. So if we I want to further clarify that all drug products, oral drug products, are not required to go through the bioequivalence studies. Only those products are required bioequivalence studies which are in the BCS classification 3 and 4 specifically, and BCS 2 class drugs are also some part of that required bioequivalence studies. So now, what is the prime parameter for the granting equivalence studies? That is called comparative dissolution profiling, which we call CDP. CDP is basically in vitro testing, which every drug manufacturer is supposed to perform to establish that its product, generic product, is equivalent to that of the innovator drug product. So we are following this CDP criteria for the equivalence studies before granting any regulatory approval. And another thing which was shared by Dr. Bhatti, that is whether the questions which he has put in the beginning at the end, and he has also answered that all generic products are by and large equivalent to in clinical efficacy 80% to 125%. And interestingly, this margin is also being allowed by the Bible of all our pharmaceuticals, that is the pharmacopoeia. If we see any pharmacopoeia, then the limits of the efficacy and quality is very clearly mentioned, plus minus 10 or plus minus 15. So we can easily and safely say that all the products which are generic or follow-up products, these products not only qualify the criteria of efficacy and quality if they follow the pharmacopoeia of like the Stated Pharmacopoeia or British Pharmacopoeia, which we are also following. Just to update 2 things to you. There is a perception that might be I'm wrong, but there is a perception that our regulatory authority is like some banana regulatory authority. So believe you me, you are working with us since long, that we are working on same lines which the international regulatory authorities are working. And just to share that in last 3 to 4 years, we have been audited by WHO. We have been audited by the United States Pharmacopoeia, and USPN has declared that Pakistan has adopted one of the robust system for the grant of the registration. I am not saying perfectly that we are we are at the height of our regulatory excellence, but definitely we are on the path of that. And for this purpose, because all these bioequivalence studies require some legal provisions. So in 2017, we have got approval of our biostudy rules as already shared by Dr. Raza Shah. And after that, this biostudy rules, now about 6 or 7 centers have been approved. Dr. Rashid will share with you. And now we are planning to adopt this bioequivalence studies as mandatory studies, so starting with the BCS level 4 and 3. And now, just to share with you that we are planning with our ministry to have national bioequivalence policy as well. Because we live in a country where if you don't have some regulatory hammer, then it's very difficult to implement. So we are very actively working on the national bioequivalence policy as well, so that we can have all of our products on the same lines and patterns with the same efficacy and quality patterns and criteria, which is supposed to be done in any standard and reference regulatory authority. And just to share with you that for this purpose, we have already adopted 19 stringent regulatory authorities of the world as our reference regulatory authority. By and large, there are 27 stringent regulatory authorities in the world, but we have established a criteria and we have shortlisted only 19 regulatory authorities as our reference authorities, and we follow same criteria for evaluating any drug product before granting registration, which they follow. So it was all about the regulatory scenario of the Pakistan, which was supposed to be shared by me. Thank you very much, Dr. Zand.
Great.
Anything
Great.
I'm available.
Yeah,
Can
Dr.
I add
Badullah.
something, Dr. Uzair?
Yes, please, sure.
Yeah. So I just want to thank Dr. Badullah. This was great that, in Pakistan, we have such stringent regulations. My just 2 cents are like that. Please make sure when you're going through all the specifications, draw specific attention to the impurities. I know the active ingredients are it's always considered, because they have all the pharmacological action. But more and more now, the know-how in our scientific community is that, there is a tendency, I'm not saying it in Pakistan or anywhere, but just in general tendency that, the impurities, which are sometimes very carcinogenic and very toxic, they are not rigorously monitored and considered. Number one comment. Number two is that, do you guys have any deadline for for making this, as you mentioned that, class 2 and 3, 3 and 4 classes of drugs as a mandatory as a bioequivalence, because the therapeutic efficacy is directly related to the release of the drug from the tablet matrices, as you can see that the solubility and the permeability, and this dictated by how the formulation is formulated due to the excipients within the tablet, even though their dissolution profile, in my experience, my humble experience, I would say that we have done like more than 700, would say that their bio dissolution profile is superimposable, but still some fails in the bioequivalent studies. That's why I offer my services. I owe to my homeland. So I get always emotional talking about Pakistan.
Yeah, just Dr. Uzair, if you permit me, I just want to respond in 1 minute. In fact, you have first talked about the impurities. Yes, you are very much right. And this impurity is not only related to active pharmaceutical ingredient impurities, rather it is also sometimes process-related impurities.
Right.
And believe you me that we are already working on this project, and at our Center Drugs Laboratory, we have already established a reference laboratory for the APIs, drug substances, and impurities as well. And in the first phase, we have implemented that all the active pharmaceutical ingredients or drug substances will be imported from the GMP licensed facilities, drug substances. And now we are moving towards the impurity profiling of the all substances as well. And second point, which you have mentioned about the BCS 3 and 4 system, definitely it will take some time until as we have some policy for the bioequivalence. But at the same time, the point which you have mentioned about the release profiling and drug matricing and all the things, because we know that it is all related to the physicochemical properties and drug particle size of the substance and matrix. So we have already implemented in vitro profiling of all such products which we are going to approve. So in vitro profiling, including particle size and resolution profiling with the innovator prevent is mandatory. But I hope that PCS class 4 and 3, and we have also started, like, because you know the prevalence of tuberculosis is very much high in the Pakistan. So we are already planning to start this bioequivalence studies with all these PCS4 and anti-tubercular drugs. But there is one problem, that is the whether it's the chicken first or the egg first. Because right now in Pakistan, we don't have some high number of pharmaceutical these bioequivalence centers like Professor Raza Shah has shared that they have the capacity to undergo all the about 110 patients, and so all these and some other centers are also there. Dr. Abdul Rashid will share, but definitely these centers are not essential to cater the national need. So we are vigorously working to establish and to facilitate all these bioequivalence centers so that we can have we can make all these requirements as a mandatory requirements for the grant of the registration. Thank you.
Thank you.
So nice of you. And Dr. Ubaid, maybe with your permission, may I request Dr. Bhatti, because he's so generous, kind, and having so much of knowledge, that maybe a series of webinar for capacity building with the DRAP people, if you would like to request him, and if he can spare some time, so that will be a great service. And we have the Dr. Abdul Rashid with us, who is the director and looking after the pharmacy service. Dr. Abdul Rashid, can you share how many labs are functional for bioequivalence in Pakistan, and what is their maturity level and the facilities available? I know we are running short of time, we are quite late, but I think this is the final thing. We must know very clearly what is the capacity available and their level and competency, please.
Thank you very much, Dr. Uzair Saab, and also the excellent presentation from Dr. Masood Bhatti Saab. I tell to everybody that there are 5 bioequivalence centers approved by the Clinical Study Committee DRAP. So these 5, among these 5, I will must mention the name of ICCBS, HJ Karachi, which the human resource is well trained from Jordan. They visited Jordan, they trained there and conducted many bioequivalence studies in their centers. After that, second one is the Olive bioequivalence centers in Lahore. That is good one. He is son of Dr. Amjad Java, that is Ali Java. He is a clinician and very good expertise in bioequivalence studies. So beside this, I will mention that Pakistani people can be confident when a generic drug product is approved by the Decision Board DRAP if it has met the rigorous standards established by DRAP with respect to identity, strength, purity, and potency. All generic drugs are put through a rigorous multi-step approval process that includes a review of scientific data on the generic drug, ingredients, performance, and effectiveness. Division of Quality Assurance and Lab Testing also conducts inspection of the manufacturing units, and all the brand and the generic medicines are manufactured in the same good manufacturing practices facilities. DRAP monitor drug quality even after the generic drug has been approved, and post-marketing services is conducted throughout the country. Generic drugs always made in the same field, as I have already mentioned, in the same name, having the same good manufacturing facility. And 3,000 inspections, GMP inspections of the industry, and have more than 50,000 samples are taken for test analysis. In India, an average of 38,000 samples are tested, but in Pakistan also the more than 38,000 samples are tested. As far as you, Dr. Uzair mentioned, the capacity of these centers, bioequivalence centers in Pakistan, it is very less. The maximum and the best facility and have the even more capacity in Karachi, other cities, and here we have conducted the trainings on bioavailability, bioequivalence, CROs, clinical trials, and also the bioanalytical laboratories with the sport USP plus, and the trainer was Dr. Sarfaz Niazi. We have 6 sessions in different hotels and also the PKM office in Islamabad. We also conducted clinical trials with the support of Comstruct. We also conducted the trainings for these centers with the support of Comstruct also supporting fund. Also, I will invite because I'm working as a chairman clinical study committee and also the director of division of pharmacy to Dr. Masood Bhatti, that he should spare some time and attend to Pakistani scientists. With this, I'll thank to everybody.
Thank you very much, Dr. Abdul Rashid. And unfortunately, we are really running short of time. We are already over by 26 minutes. We do have the miscellaneous question. My sincere apology to the people, but we will try to respond to your question through emails. And with that, I think, Dr. Fraser, do you have something to say or add, please? Final word.
Thank you, Dr. Uzair. Very, very informative and very valuable presentation by Dr. Masood. I have one brief question. What do you think, where do we stand ethically in a condition, for example, if it's a patient of a cancer, and we know, and as you also mentioned this, that there might be some like a difference in effect by 5 to 10% between generic and the branded drug. So would you still prescribe a medicine to a cancer patient, a highly risk cancer patient, a generic drug? Keeping in mind this, that 5 to 10% effect can be a game changer.
Okay, Dr. Masood Bhatti, please.
You know, there has been a series of discussions about that, and you know, it has to be decided case by case basis. I don't think we can make any generic general assessment for that. It is always patient's right first. So, you know, all the ethical board, you start from the Helsinki Accords to any type of GCP guideline. So patient rights come first. So this is our almost responsibility, that whatever we do, it has to be in the best interest of the patient. And at the ground, at the time, if the physician and the experts, medical experts, if they consider, they think that nothing else is available and there is the only way they can save and benefit this patient, then they can make that decision. As I said, it has to be as a case by case basis. And if you feel that it is in the best interest of the patient, then the medical experts can make their judgment there.
Dr. Masood Bhatti, can you add something about clinical equivalence?
Clinical equivalences, right? So, you know, there's a pharmacodynamic studies are we talking about for the drugs which are not solid dosage form, or general clinical equivalence or therapeutic equivalence. So there are, for example, biologics, they're called biosimilars. So they are There's something like generic drugs, but then it has another whole science behind it, how we do biosimilars. So I did not touch that topic, but clinical equivalents are when you do pharmacodynamic studies where you cannot have enough levels in the plasma to compare the AUC. So then you have to do what we call it a pharmacodynamical clinical endpoint studies to show the equivalency.
Thank you. I want to add something for the participants that we have conducted
I think there is a problem with the signals from Dr. Abdul Rashid.
That allopathic medicine
Dr. Abdul Rashid
should be prescribed by in generic name.
I'm sorry, you are not clearly heard because there is a signal issue from your side, so he cannot understand the question. But anyway, you can write to us and we will try to forward it to Dr. Masood Bhatti for detailed clarification. With all this, I think we are quite about half an hour over the time. But thank you very much, Dr. Masood Bhatti. Trust me, that's one of the fantastic, very knowledgeable presentation. And let's talk about activity. And we learned a lot on behalf of APPNA Merit and the participant. We are thankful to you for such a great talk. And I'm also thankful to the industry people who participated in today's session, because that's very, and once again, apology to the people who asked questions, but because of the time, we could not take it. So with this, I say goodbye to Dr. Masood Bhatti, Dr. Abdul Rashid, Dr. Ubaidullah, Dr. Fasid Zaman, Professor Dr. Muhammad Raza. Thank you very much for your participation, and we will continue. And have a great afternoon or night, good night, or whatever, wherever you are. Thank you again to everybody.
Thank you so much. Thank you to everybody, including APPNA Merit, Walid Zafar, all the participants, panelists. Thank you so much. Have a wonderful evening, afternoon. Bye now.
Great.
Assalamu alaikum, everybody.